So this is a review of the course, the topics that we covered, the topics that are game for the final exam. So we covered respiratory, the chapters on respiratory, chapter 22, blood vessels, heart, lymphatics, immunity, urinary systems, fluid, electrolyte, and acid base, reproductive systems. I believe that that's about 10 chapters. I didn't actually count them up, but it means that there are between kind of 8 to 10 questions out of each chapter. Obviously, the immunity chapter we spent more time on, so there will be more questions on. So expect that there will be about 8 questions to 10 questions per system, approximately. So in chapter 22, the respiratory system, we looked at the anatomy of the respiratory system, so the nose and the sinuses, the pharynx, the larynx, the trachea, the bronchi and the subdivisions of the bronchi, the lungs, the pleura, and that kind of stuff. We talked about the function, and as you from last class and like last semester and this semester, every one of these chapters starts out with what is the function. So in for respiratory, the function is to supply the body with oxygen and get rid of the volatile gases like carbon dioxide, the waste stuff. And the functions, there's 4 processes has to happen: pulmonary ventilation, external respiration, transport of the gases, internal respiration, all has to happen. So we divide it into the respiratory zone, the place where this external respiration happens, and the conducting zone, which is really involved with ventilation. So we looked at the epithelium and all of that. The larynx, the trachea, vocal cords, how sound is developed. The special features of the trachea, like the horseshoe rings. We looked at the bronchi, right down to the terminal bronchioles as the end of the conducting zone. Then the respiratory bronchioles, the alveoli and how they are. We looked at the lungs and the pleura. How many lobes are in each lung? What are the regions of the gross anatomy of the lungs? What's the histology of the lungs? When we looked at the individual alveoli, we talked about the respiratory membrane being the type 1 cells of the alveoli and the walls of the capillaries and their fused basal lamina. We talked about type 2 cells making surfactants. We talked about macrophages in there, keeping things cleaned out. We got into the mechanics of breathing, so the physiology of ventilation, so the pressures in the thoracic cavity, intrapulmonary pressure, intrapleural pressure, transpulmonary pressure, that kind of stuff. We examined Boyle's law, and so I'm expecting that you should be able to talk about Boyle's law. We talked about inspiration being work and expiration, usually at least passive expiration is a passive process. we looked at the factors that affect it, so airway resistance, the surface tension of the alveoli, lung compliance, so scarring and that kind of thing. We then talked about respiratory volumes. So know the residual volume, the expiratory reserve, the tidal volume, the inspiratory reserve, the functional residual capacity, the inspiratory capacity, the vital capacity, the total lung capacity. You don't have to know the numbers, but you have to know what that's about. This table 22.2 on page 823 looks at the effects of breathing rates, alveolar ventilation versus total lung ventilation. It's the idea of dead space. Non-respiratory air movement, so laughing and coughing, sneezing, crying, hiccups, that kind of stuff. Then we talked about the physiology of exchange, of external respiration. So Dalton's law, Henry's law, the composition of alveolar gas, thickness of the membranes, ideas of partial pressures, gas solubilities, ventilation perfusion coupling. We briefly touched on internal respiration. We talked about how oxygen is transported on hemoglobin, how carbon dioxide is transported. 7 to 10% is dissolved in plasma, 20% is bound to hemoglobin, and the rest is bicarbonate. And we saw later in the course that bicarbonate matters a lot to pH. We talked a little bit about the Haldane effect and that kind of thing. Control of respiration, being in the medulla and the pons, so it's all brainstem stuff. There are chemical factors, so the influence of partial pressure of carbon dioxide is the highest stimulus, so hypercapnia it's called. The partial pressure of oxygen lowering, it has to get very low before it carries on. There are higher brain centers control on that. And we talked about inflation reflex, and I think that's pretty much where we ended this chapter. The next chapter we did was chapter 17 on blood. We looked at an overview of blood, the components of blood, the physical characteristics of blood, the pH, ideas of what centrifuge blood looks like, the plasma being 55% buffy coat, and the hematocrit, being 45%, which is red blood cells. pH 7.35 to 7.45. There's about 5 liters of blood in the average person, 5 to 6 in men, 4 to 5 in women. But it really has got to do with more body size than sex. We looked at blood plasma. The constituents of blood plasma, 90% of it's water, the electrolytes, plasma proteins. The 3 proteins that are really important are the albumin, the globulins, and the fibrinogen. There's nutrients, respiratory gases, hormones, all found dissolved in the blood plasma. We looked then at the formed elements, the red blood cells, the size of them, the shape of them, that biconcave disc. We looked at functions of red blood cells, and therefore hemoglobin. We talked about erythropoiesis, where they come from in the bone marrow, the hemocytoblasts, which are the hemopoietic stem cells, and eventually becoming erythrocytes. How we regulate erythropoiesis with the hormone erythropoietin, which comes from the kidneys. We looked at what you need in the diet. And then we looked at what happens to the erythrocytes because they can't repair themselves. We looked at their lifespan. We spent some time looking at the ultimate fate of it. Then we got into the white blood cells. They never let monkeys eat bananas. The granulocytes versus the agranulocytes. The granulocytes all have fill in it, so they're neutrophil, eosinophil, basophil. The agranulocytes are lymphocytes and monocytes. Kind of what each one of them is used for. Where did they come from? Platelets. We talked about platelets and what they're for and how they're used, which got us into hemostasis and blood clotting. So the first step, vascular spasm. Second step, platelet plug formation. Third step is clotting, coagulation. So what are the two pathways, intrinsic and extrinsic pathway of getting to prothrombin activator? And then prothrombin becomes thrombin, and thrombin causes fibrinogen to become fibrin, and clotting happens. Then clot retraction happens. And then later fibrinolysis happens to remove the clot, but that's considered a second thing. We talked about the ABO and RH blood groups, the self-antigens in transfusion reactions. So that was that chapter on blood. Chapter 18 is on the heart. We looked at the heart anatomy, the size, the location. We looked at the two circuits, the pulmonary circuit and the systemic circuit of circulation, and how the heart relates to it. We looked at the covering of the heart. So we looked at the pericardium, the fibrous pericardium, the serous pericardium, so the parietal layer of the serous pericardium, the pericardial cavity, the epicardium, the myocardium, the endocardium, the lumen of the heart. So those are the layers of the walls. We talked about the chambers, the four chambers, the two atria, the two ventricle, the interatrial septum, the interventricular septum, the coronary, the surface, so the coronary sulcus, the anterior interventricular sulcus, the posterior interventricular sulcus. We talked about the valves, the atrioventricular valves versus the semilunar valves into the great vessels. We named them. We talked about how the valves were backed up with chordae tendinae and how they worked. We spent a very brief amount of time on coronary circulation, so the coronary arteries, and naming the coronary arteries and what they're for. We looked at cardiac muscle fibers and the striation, the intercalated discs, that kind of thing, all the mitochondria. We looked at we spent a lot of time on the heart physiology, so we looked at the electrical events of this. So this intrinsic cardiac conduction system, I spent a good deal of time on, looking at the SA node, the AV node, the AV bundle, the bundle of His, the bundle branches, the Purkinje fibers, and how they work and how they set the rhythm of the heart. So this is all on page 674, 675. Looking at that. We talked about how we modify the basic rhythm so that uh, the SA node would set it for about 100 beats a minute. We use the vagus nerve and the parasympathetics to slow it down to its normal resting rate, and then we inhibit the parasympathetics to increase the heart rate, and we use the sympathetics to increase it even further. We looked at the electrocardiograph, so the PQRS, and what happens there at each one. We talked about the mechanical events of the heart. So we looked at diastole and systole, and so we looked at ventricular filling, ventricular systole, the isovolumetric relaxation, isovolumetric contraction, that all of that stuff we looked at. We talked about cardiac output, and so stroke volume times heart rate, and what are the things that affect stroke volume: preload, contractility, afterload. Frank Starling's law of the heart. I spent a good deal of time talking about that. So regulating cardiac output by regulating stroke volume. Because we then talked about how to regulate cardiac output by regulating heart rate, and that's really the autonomic nervous system. We didn't talk about the developmental aspects or any of that. We talked about valves and heart sounds, and went from there. The next chapter we did, chapter 19, cardiovascular system, it's the blood vessels. We spent the majority of the time looking at the structure and function of blood vessels. What's an artery? What's a vein? What's a capillary? What are they used for? What are the walls of these blood vessels? So the tunica intima, which is also known as the tunica interna, the tunica media, with the muscles, vasoconstriction, vasodilation, tunica externa or adventitia, the vasa vasorum. So all on page 693, 694. What are they for? The different types of arteries, elastic arteries versus distributing arteries, muscular arteries, arterioles, capillaries, the different types of capillaries, capillary beds and how they work, veins, venules, veins, the compliance, blood reservoirs, 60% of blood at any given time is in the veins. On page 699, figure 19.5 is the relative proportion. What's an anastomosis? What causes blood flow? So we started talking about the physiology of circulation. So blood flow is blood pressure versus resistance. Resistance is caused by blood viscosity, blood vessel length, blood vessel diameter, and there's a relationship between these. So flow is directly proportional to delta P, and it's inversely proportional to resistance. We looked at mean arterial pressure, just the idea of blood pressure, systemic blood pressure, and how that's maintained. With neurological controls, neural controls, hormonal controls. We talked a little bit about pulses. We looked at velocity of blood flow. Blood flows fastest in the aorta and slowest in the capillaries because of the total cross-sectional area. We touched on blood in different areas. We looked at bulk flow, like On page 716, we looked at the exchange of gases, and so hydrostatic versus osmotic pressure, and what causes that. Then we just briefly touched on the principal vessels of circulation. I'm not going to be putting any emphasis on the exam of this stuff, the names of the specific arteries and veins, for the rest of chapter 19. Chapter 20, the lymphatic system, lymphoid organs and tissues. So we looked at lymph flow, the functions of lymphatic, where we find lymph capillaries, etc. So we look at lymph capillaries, the mini flaps, the permeability. We go capillaries to collecting vessels, through larger lymph vessels, into trunks, and then ducts. The names of the trunks and ducts and where they end up, and how they end up back in the subclavian. How does lymph move? The lymphoid tissue, that's reticular connective tissue. Lymph nodes, more afferent than efferent, etc. Other lymphoid organs such as spleen, tonsils, thymus, mucosal associated lymphatic tissue. Chapter 21 is the immune system, innate and adaptive body defenses. So know the difference between the innate and the adaptive defenses. The innate defenses, the surface barriers, skin and mucosa, acid mantle, enzymes, mucin, the defensins, other chemicals, that kind of stuff. The innate defenses, so cells, phagocytes, natural killer cells, processes, inflammation, fever, and as part of inflammation, phagocyte mobilization, so chemotaxis, diapedesis, margination, so kind of the leukocytosis, cytosis rather. Stuff, antimicrobial proteins, interferons, complement, what does complement do for us, that kind of stuff. Then we did the adaptive defenses, specific, systemic, it has memory, it's based on antigens, complete antigens, immunogenicity, reactivity, haptens, which are incomplete antigens, they have reactivity but not the immunogenicity. What's an antigenic determinant? MHC, self-antigens. Where do lymphocytes come from? Antigen-presenting cells. So what's the point of antigen-presenting cells, and what cells can do it? The humoral response, so activation of B lymphocyte, cloning, the development of plasma cells and memory B cells. The plasma cells make the antibodies. Primary and secondary humoral responses, how they work. Active and passive humoral immunity. So passive is you acquire the antibodies, active you make the antibodies, natural is a natural process, artificial is some sort of a drug or a needle. Antibodies, what do they do? What are the 5 classes of antibodies? So neutralization, agglutination, precipitation, complement, to fix and activate complement. So enhancing phagocytosis, inflammation, cellular lysis is what complement does. MADGE, the cellular immune response, so the difference between the class 1 and the class 2 MHCs, CD4 and CD8s, the antigen binding, the idea of co-stimulation, what are the effector cells, so what's a helper T cell do, what does the cytotoxic T cell do. We only barely touched on the regulatory ones. We looked at the chart that brought all of this immune together. We didn't do any of the homeostatic imbalances, autoimmune, or immune insufficiency. So, obviously we spent 2 weeks doing immunity. There's a fair amount on the exam. Chapter 23, the digestive system. We looked at digestive processes, so that's what takes place in the alimentary canal, the digestive tract, GI tract. So we looked at ingestion, propulsion, mechanical breakdown, chemical breakdown, which is digestion, absorption, defecation. We looked at the difference between the organs of the digestive tract and the accessory organs. We examined the mesenteries and the peritoneum and the visceral and parietal peritoneum. We looked at the alimentary canal layers, so the mucosa, submucosa, muscularis, and serosa, and what happens in each of those layers. The mouth, we talked about the tongue, the lips and cheeks, the taste buds and things on the tongue, the salivary glands, the names of the various salivary glands, the composition of saliva, control of saliva. And what I said about teeth was know the number, don't worry about tooth structure. Esophagus, the type of epithelium, the type of musculature. The top part is a skeletal muscle, the bottom part is smooth muscle, in between it's a mixture. The stomach, gross anatomy of the stomach, the fundus, the cardia, the body, the pylorus, greater and lesser curvature, the rugae, the walls of it, the microscopic anatomy, so the cells of the gastric pits, the chief cells, the parietal cells, the mucous neck cells, the enteroendocrine cells, what they all do. Regulation of gastric secretion, cephalic phase, what's in your head. Gastric phase is when food's in the stomach. And intestinal phase, the inhibitory phase, when the duodenum starts to get the chyme, the making of the chyme, the idea of intrinsic factor, vitamin B12, small intestine, the gross anatomy of it, duodenum, jejunum, ileum, plicae circularis, villi, microvilli, all for surface area, just gross anatomy of liver, the gallbladder, the ducts for it. Composition of pancreatic juice, what the pancreas does for us. The segmentation versus peristalsis in the small intestine, the large intestine, subdivisions of it, cecum, ascending, hepatic flexure, transverse colon, descending colon, transverse colon, then the splenic flexure, then descending colon, sigmoid colon, rectum, anal canal, anus, anal valves, that kind of stuff. What goes on in the large intestine, making poo, really. Digestion, chemical digestion being hydrolysis, absorption, the brush border enzymes, things like that, water absorption, and that's it for the digestive system. The urinary system, chapter 25, the kidneys, they filter 200 liters of fluid a day. We piss about 1.5 liters, so most of it's reabsorbed. What does the kidneys do for us? They regulate the total volume of water in the body, concentration of solutes, the concentration of ions, the acid base, they excrete metabolic wastes, produce erythropoietin, renin, convert vitamin D to its active form, a little bit of gluconeogenesis. Right? The different parts, the kidneys, the ureters, the bladder, urethra. So the external anatomy of kidneys, the renal fascia versus the fat adipose capsule and the fibrous capsule, or the renal capsule it's called. Internal anatomy, so the renal medulla, the renal cortex, the pyramids, the papilla, the minor calyces, the major calyces, the pelvis, blood supply, so aorta to renal artery to segmental to interlobar to arcuate to the cortical radiate, afferent arterial, glomerulus, efferent arterial, peritubular, and then back through the veins. Nephron, the structural and functional unit, the renal corpuscle, the glomerulus, the Bowman's capsule, what happens here, filtration, the way the podocytes work, the filtration slits, the renal tubules, the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, the collecting duct. This is where urine is actually produced. Very good to be able to identify all the parts of a typical nephron. The peritubular capillaries, the vasorecta, if it's a juxtaglomerular or a juxtamedullary nephron. All of this is 960. The diagram is 961. The juxtaglomerular apparatus, what's it for? How does it work? The idea of filtration and filtration rate, what's the filtration membrane, the pressures that affect filtration. So like I say, the GFR, renal autoregulation, I talked about. Reabsorption, what gets reabsorbed where, reabsorption of sodium, primary active transport, reabsorption of nutrients would tend to be secondary active transport. Water goes by osmosis. Kind of what goes where. The idea of the transport maximum, that there's only so much can be facilitated, so that you reach the max. And tubular secretion, just definition of it. So it's most important with potassium. That really, out of the regulation of urine concentration and volume, it's hormonally controlled. That antidiuretic hormone makes you make more concentrated urine. Atrial natriuretic peptide makes you make less concentrated urine, and it does it by opening the aquaporons in the opening or closing aquaporins in the distal convoluted tubule and the collecting duct. What does aldosterone do? Increases sodium reabsorption, potassium secretion. Don't worry about all of the countercurrent multipliers and all of that stuff. I didn't do renal clearance. Wouldn't hurt to look at the physical characteristics of urine, but basically that it's acidic, and that it's mostly water. There's uric acid, nitrogenous waste, and that kind of thing. I did a very short video or YouTube on the ureters, the 3 layers of the ureters, peristalsis, urinary bladder, the trigone, the rugae, the detrusor muscle, urethra, and urination, micturition. How does that happen? So that's the urinary chapter. The fluid electrolyte acid base chapter, chapter 26. It's body fluids, intracellular versus extracellular. Know the proportions of that, the compartments, electrolytes versus non-electrolytes. Which are the major cations in the intracellular, which are the major in the extracellular, sodium. Movement being mostly osmosis and hydrostatic pressure. Major sources of water intake and output. So we get it from metabolism, 10%, foods give us 30%, beverages give us 60%. We lose it in feces, sweat, breathing, and urine. So thirst, how it works, regulation of water output, and antidiuretic hormone, electrolytes, aldosterone, sodium, and potassium. So aldosterone, atrial natriuretic peptide are the big things that affect it. Other ones would be progesterone, estrogen, the glucocorticoids. Same with potassium, regulation of calcium, parathyroid hormone, calcitonin, calciferol. Acid base, chemical buffers, bicarbonate, weak acid, strong acid, phosphates, protein buffers, respiratory regulation of hydrogen ions. So we breathe away if we have excess. No matter what, we'll compensate for metabolic acidosis by respiratory methods if we are trying to reach the balance. So if we're in metabolic acidosis, we will breathe rapidly to compensate, which would... Sorry about that. if somebody's breathing rapidly and they're not in respiratory acid or in metabolic acidosis, they will go into a respiratory alkalosis. So we looked at how breathing does that, and we looked at the renal mechanisms. So first you try and neutralize the acid, then you try and breathe away any excess acid, then you pee it away. So the different types of acidosis and alkalosis. And that was it for chapter 26. Chapter 27, reproductive system. In class we did female. On the YouTubes we did male. So with the male we talked about the gonads being the testes, the secondary organs being, the accessory organs being the penis, and primarily, and the various glands. We looked at the scrotum, the spermatic cord, the dartos muscle, the cremaster muscle, testes. We looked at the seminiferous tubules, the ready testi, the efferent ductules, the epididymis, the vas deferens, where that all runs, the testicular arteries and the pampiniform venous plexus, about maintaining temperatures, the penis, the corpus cavernosa, the two of them, the corpus spongiosum, the various arteries, the duct system, so the epididymis, vas deferens, the urethra, the seminal vesicles, what do they do? The prostate, what does it do for us? Bulbourethral glands, what do they do? Semen, what's that about as opposed to sperm? What causes erection? What causes ejaculation? Spermatogenesis. I talked about meiosis and making 4 haploid cells out of 1 diploid. That this is done in the seminiferous tubules. I said don't worry about each stage, the primary spermatocyte and secondary spermatocytes and early spermatids and late spermatids. The important thing is that we end up with 4 spermatozoa, 4 sperm cells for each really stem cell. There's hormonal regulation, so what happens with the FSH and LH and testosterone. Secondary male characteristics. In the female reproductive system, we looked at ovaries being the gonads, that make the gametes, and the sex hormones, estrogen and progesterone. So we looked at the ovaries, where you find them, how they're supported, the mesovarium, suspensory ligaments, the tunica albuginea, the fact that there is a cortex and a medulla. We looked at the duct system, the fallopian tubes, so the infundibulum, the ampulla, the uterine tube, the isthmus, the uterus, the body of the uterus, the fundus of the uterus, the uterine wall, the perimetrium, the myometrium, and the endometrium. support of the uterus, the vagina, so the external genitalia, so the entire vulva, the mons pubis, the labia majora, the labia minora, greater vestibular glands and why they're there, the clitoris, the glands, the prepuce, the body of the clitoris. We didn't really discuss mammary glands, but basically mammary glands are modified sweat glands that produce milk. They are fats in non-lactating women, and really they're primarily decorative. It's because humans have what's called occult ovulation. Almost all other animals, including all the other primates, breasts are only present when there's lactation happening. With males or with humans, it's different. We talked about the physiology of the female reproductive system, so the whole idea of oogenesis, primordial follicles becoming primary follicles, becoming secondary vesicular follicles, ovulation, corpus luteum, what's happening with the oocytes through that, the polar bodies, so the whole ovarian cycle, what's happening in the ovaries. We looked at the picture on page 1046, looking at the different types of follicles. As we talked about the different phases of the ovarian cycle, so there's the follicular phase, the ovulation, and then the luteal phase. What are the hormones? So in the follicular phase, it's estrogen. In the luteal phase, it's progesterone. The uterine cycle, the uterus is a separate organ, so it has its own cycle. So the first part is the menstrual phase. That happens when there is virtually no progesterone or estrogen, so the hormones are very low. Then that's followed by a proliferative phase where the endometrium is rebuilding and growing. And then that's followed by the secretory phase, and that's normally the secretory phase happens right after ovulation. And in the secretory phase, the uterus is preparing for pregnancy. It's ready to retain, receive, and nourish a fertilized ova. We looked at what was happening in the pituitary, so the FSH and the LH, and how that relates to what's happening in the ovarian cycle and producing estrogen and progesterone, and how that controls what's happening in the menstrual cycle for menstruation, proliferative phase, and secretory phase, and how these things all have to basically work together to be for the betterment of the species, as it were. The effects of estrogen and progesterone, female sexual response. We didn't look at the STIs or the developmental aspects of reproductive system at all. So the exam, like I say, covers roughly equally everything. A little bit less on the chapter of blood vessels, maybe a little bit less on the lymphatics, a little bit more on immunity. a little bit more actually on the reproductive because some of that wasn't tested. I said there are 3 diagrams. One of the diagrams comes out of the urinary chapter. It's and it's a matching, so you match the letter on the diagram to the number of the question. One of them is a reproductive, so you match letters and numbers, and the last one is cardiovascular. It's heart, and it's again a matching. 3 hours and 100 questions. See you Tuesday.